Hi guys, this is Mr. Sodal, and on today's OV we're going to be learning about mutations on both the gene and chromosomal levels. Mutation is an often misunderstood term. Not all mutations are bad. In fact, the great variety of life on Earth is all due to mutations in the DNA of living things. Today's PowerPoint, the first thing we're going to talk about is what causes mutations. Mutations are caused by spontaneous errors that occur during DNA replication. Most of these errors are caught and repaired by the enzyme DNA polymerase in its proofreading function during DNA replication. However, not all of these errors are found, and the ones that are missed are called mutations and can lead to changes in the expression of this DNA. There are some environmental factors that can increase the chance of mutations happening. And the two factors that we're going to look at are radiation and certain chemicals. Radiation is the process by which electromagnetic energy travels through space or a medium such as air. An example of a mutation causing radiation is ultraviolet radiation. And really any of your higher energy radiations can cause mutations, such as gamma rays or even X-rays. They could lead to such things as cancer. It can also, these ionized molecules can cause mutation in DNA, can lead to cancer, as I mentioned, the rapid growth of your cells. Some chemicals that can cause mutations cause nucleotide substitutions, which we will learn more about in a few minutes, and they change the number of nucleotides in the DNA. Nitrates into nitrous acid and other chemicals that are used in food preservation can lead to these mutations. Some other chemicals structurally change the bases in the DNA, causing a base pair change after replication. For instance, 5' bromouracil structure, shaped just like thymine, inserts itself and results in a base pair changing G to C into A to T. Basically, this chemical, this 5-bromouracil, takes the place of thymine, and instead of having a TA pairing, a GC pairing inserts. And believe it or not, just that one little change in base sequence can lead to big changes in protein and function for the cell. So the first type of mutation that we're going to learn about is called a gene mutation. Now this is a category of mutations, and it's a change, it's very important here, a change in just a single nucleotide in the sequence. One single nitrogen base gets changed in a gene mutation. There are two types of gene mutations. The first type are called point mutations, meaning they happen at a single or very few points within the DNA sequence. Mutations that involve replacing one or more nucleotides in a DNA molecule with another incorrect nucleotide. It affects only a single codon and therefore usually a single amino acid. And an example would be a substitution, where one base is substituted for another. Another example of a gene mutation is a frame shift mutation. These have a little bit bigger impact on the DNA and then the protein. This is a mutational event in which a number of bases other than multiples of 3 are added to or removed from the DNA, causing a shift in the codon reading frame. Okay, and we'll learn more specifically what that means in the next few slides. And examples here are called deletion, where a base is removed, or insertion, when a base is added mutations. Substitution point mutations. There are 3 types of substitution point mutations that we need to be aware of. The first type is called a missense mutation. A missense mutation alters a single amino acid. Okay, and it does this when one or more nitrogen base are removed and replaced by another incorrect nitrogen base. This does not change the overall number of nitrogen bases. We still have the same number of letters in our DNA molecule. Okay, And this can be seen using this sentence as an example. If this was the normal DNA sequence with our codons here, the fat cat ate his hat. A missense mutation, single point, the fat, now look, we changed the C to an R, the fat rat ate his hat. Okay, we just changed one letter, but it changes that entire codon. Therefore, it would change the entire amino acid and affect the function of this entire protein. The sentence is different now. It's about a rat eating his hat, not a cat. Okay? Another example would be multiple point mutations where all three letters are changed, still affecting just one codon, one amino acid, but it does change the meaning of the sentence. The two other types of substitution mutations are called silent and nonsense. A silent mutation occurs if the change in the DNA nucleotide does not change the amino acid sequence, meaning an A gets changed with a T, but the amino acid stays the same. Okay, as we learned about with our genetic code, there are multiple codons that code for the same amino acid. For example, the stop codon, UGA and UAA, they both mean stop. So if you replace the G with the A, it doesn't change the overall effect of that codon. It still produces a stop codon, the same amino acid. A nonsense mutation occurs if the change in the DNA nucleotide inserts an extra stop codon prematurely or too soon and ends the DNA sequence, which therefore would shorten the protein, which is not good. Okay, so basically adding a stop codon in the middle of the DNA sequence, okay, which would then cut the protein short and therefore prevent it from functioning properly. An example of a substitution mutation. Okay, it says here, create a substitution mutation by switching the third thymine to a cytosine. What change in the amino acid chain has been made? So if we look here for our DNA molecule, we have our codons TAC, ACG, GAT, CCC, CCC, ATC. Notice normally it should be CTC and then 3 Cs. If we follow this error, this substitution of a C for a T, we see that now instead of GAG, it will be GGG, and instead of producing glutamic acid as our amino acid, we produce glycine. Okay, and believe it or not, this amino acid, this protein, will function differently, will have a different shape than the normal protein, even though only one amino acid is different. And that could potentially affect its structure. This would be an example of a missense substitution mutation. One base has been changed from the normal, resulting in one amino acid being changed. If this was a nonsense mutation, instead of producing glycine, we would produce a stop codon here. If it was a silent mutation, this change would have still given us a glutamic acid. Okay, but that's not the case. This is a missense mutation where the amino acid sequence has been changed by one. Frame shift mutations, as I mentioned before, are a little more impactful. They have a greater effect on the protein and can actually cause greater damage and loss of function for the proteins. Frame shift, again, are your deletions and your insertions, where we either delete a nucleotide or add an extra nucleotide. This causes a change in the reading frame of the codon. When we look at an example in a few minutes, you'll see what I mean. This often produces drastic alterations or dramatic alterations in the structure and function of the protein, as I mentioned. And note, how many amino acids are now going to be affected? Remember, in our point mutations or substitutions, we only affected one amino acid. In a frame shift mutation, we affect every amino acid after the mutation. Okay, so it has a greater impact. So again, here is our normal sentence. The fat cat ate his hat. If we have a frame shift and we added an extra A here, notice what happened. Here's our insertion. Now it's the fa, ta, ka, tat, ihai, sha, ti. All right? The whole sentence is gibberish after the mutation. We've added an extra A. We see we have an extra nucleotide here. No longer are groups of 3 for our codons. The reading frame has shifted. Okay? We can no longer read the word fat cat ate his hat. Okay? It pushes everything to the right because we've added an extra nucleotide. The opposite happens in a deletion. If we delete the A, now it's the futka ata te ish at. All right, again, gibberish. All right, because all of the amino acids shifted to the left. Excuse me, not amino acids. All of the nucleotides shifted to the left. Okay, and now since we have to read in groups of 3, that's our codon, have to read in groups of 3, the T and the C got pushed over. Okay, and now it's FTC, ATA as our codons. Okay, our reading frame has shifted, and we're left with not even a full codon here at the end. So we're going to be 1 amino acid shorter than we should have been in this example with a deletion mutation where we remove a nucleotide. If we look at a real example, okay, here's an example of a deletion frame shift. Okay, so here we're going to be looking, all right, at what happens when we delete the second G and what change the amino acid chain has been made. Okay, so here's our normal sequence, TAC, ACG, GAT. Okay, so we're getting rid of this G. Okay, notice ATC got moved over, TCC got moved over, CCA got moved over. I'll use a little drawing here. See if it'll actually work for me. And okay, everything shifted over. Okay, to fill the gap created by eliminating that G. So now our third codon, instead of being GAT, is ATC. That's C moved over. TCC, CCA, and then TC left over. UAG, AGG, GGU, AG. Notice what happened to our amino acid sequence. We start with methionine, cysteine. Now we have a stop codon instead of leucine. 2 glycines afterwards, and then a nothing. That's what that little squiggle is representing. This won't even create an amino acid. And notice our protein here now is just 2 amino acids and then 2 more amino acids, where it should be 1, 2, 3, 4, 5 amino acids before the stop codon. So this frame shift really drastically changed our amino acid sequence and therefore protein, which would obviously affect the function of that protein. Good news here is mutations and DNA damage can be repaired. Okay, we talked a little bit about that at the beginning of this presentation. Cells do have enzyme systems that repair the DNA. Okay, if you remember, that was DNA polymerase that proofreads the replication to fix some of these errors. The errors are corrected during replication. Okay, DNA replication, S phase of interphase. And again, DNA polymerase is the enzyme that corrects the mistakes in the DNA replication. It has that proofreading function. Go back here a second. So here's our original DNA in our diagram here, CAT, GTA. Okay, DNA is replicated, proofreading enzymes detect the mistake and make a substitution for it. All right, so we could have a mistake here and correct that. Now we're going to talk about chromosomal mutations. These are a much bigger deal than gene mutations. We'll see why here in a second. There are 2 main types of chromosomal mutations. You can have a change in chromosomal number, which is called a non-disjunction. There's 2 types of non-disjunction, which we'll talk about, polyploidy or aneuploidy, and a change in chromosomal arrangement, insertion, deletion, inversion, translocation. But as you'll see as we go through here, these are not just changes in single nucleotides within the sequence. These are changes in entire genes or entire chromosomes. So you're either missing or have an extra or have a screwed up order of an entire gene or several genes. Entire segments of chromosomes are missing or added. Or you have an entire extra set of chromosomes, okay, as we'll see in a few minutes. All right, so these can have a much greater impact. Some of these mutations will result in like a stillbirth or something, the offspring not surviving through growth and development. Non-disjunction is the first type of chromosomal mutation we'll talk about. Literally means not coming apart. Non-disjunction. Junction means sort of coming together. Dis means not coming together. Okay, non-disjunction, not coming together. This is a result of the failure of chromosome pairs separating properly during meiosis. So during anaphase, anaphase 1 and anaphase 2, either the tetrads not separating properly or the sister chromatids in anaphase 2 not separating properly. The result here, is caused by the homologous chromosomes not separating in meiosis 1 or the sister chromatids not separating in meiosis 2 or mitosis, and it results in cells with an imbalance of chromosomes. They might either have too many or too few chromosomes in the gametes. So a sperm cell with 24 chromosomes or 22 chromosomes, egg cell with an extra chromosome or missing a chromosome as well. That is the result of a non-disjunction. So we see in this diagram here in our non-disjunction, here is our meiosis 1, metaphase 1. We see our tetrads lined up together. In anaphase 1, notice this tetrad did not separate. So they both went to this side of the cell and ended up in this daughter cell. This daughter cell each of these daughter cells should have 2 chromosomes in it. This one only has 1. This one has 3, which results in an imbalance in our final 4 daughter cells. These are our haploid number plus 1, so they have 1 extra chromosome. These cells are the haploid chromosome number minus 1, only 1 chromosome. They should have 2. All right, and that's called, due to non-disjunction happening here during anaphase, the failure to separate. We see non-disjunction down here. Polyploidy is the term for when a cell has an extra copy of every single chromosome. Okay? A triploidy is a common example of this, chromosome number in which there are 3 copies of all autosomes and 3 sex chromosomes. And we'll look at a karyotype here momentarily. But what that means is instead of just having 46 chromosomes, you have an extra 23 on top of that, an extra copy of each chromosome. All right, and the karyotype would look like this: 3 chromosomes when we normally should just have 2 of each of these chromosomes, of each of these pairs. Okay, so this would be a female who has 3 X's and 3 of everything else. Okay, that would be called a triploidy, polyploidy. More common type of non-disjunction is this aneuploidy, where instead of having an entire extra set or an entire missing set, just 1 or 2 of the chromosomes have an extra copy or a missing copy. So we see here a chromosomal number that results in more or less than 46. It only affects 1 chromosomal pair, not every chromosomal pair. A monosomy, condition in which 1 member of a chromosome pair is missing, or a trisomy, where we have an extra copy of 1 of the chromosomes. So mono means you just have 1, trisomy means you have 3 of them. An example of a trisomy is trisomy 21, Down syndrome, a very common genetic disorder caused by an extra chromosome number 21. Okay, an aneuploidy involving the presence of an extra copy of chromosome 21 results in Down syndrome. This is the only autosomal trisomy that allows survival into adulthood. There's other examples, but those organisms that have this mutation, this non-disjunction, typically die either before birth, during birth, or shortly thereafter. Again, here would be a karyotype of someone with Down syndrome. Okay, it's a female, so she has 2 X chromosomes, and here's her chromosome 21 with the 3 pairs. Okay, and that would be where we have our trisomy. Okay, all the other chromosomes are normal, doubles of each. They just have a little extra chromosome 21. Notice it's a small chromosome, so there's not many genes they have an extra copy of, but it still has a great impact on the physical expression of the traits controlled by that chromosome. The change in chromosomal arrangement, the translocations, duplications, inversions, and deletions. These are a little different than non-disjunction because they are not extra or missing entire chromosomes, entire DNA molecules, but these are entire sections of chromosomes that are either missing or misplaced or extra. Okay? And again, like I said before, for this section, it's important to remember, okay, that entire genes may be missing, duplicated, or misplaced here, not just one or two nucleotides. Okay, so we're not just talking about adenine, guanine, cytosine, thymine being misplaced here. We're talking about entire genes which contain thousands of nucleotides that are either missing or doubled or inverted. First example here is a duplication. Okay, we look here, we see this is our gene sequence. So remember, this is gene A here. So this is 1,000 nucleotides long, another 1,000, another, and so forth. Okay, on the normal chromosome, it's A, B, C, D, E, F, G. Okay, if we repeat a segment, see the DE is repeated, okay, that is our duplication. It can happen a couple times. Extra copies of these genes D and gene E. All right, we don't need extra copies of genes, so that could lead to some negative effects. We do have to remember, not all mutations are negative. Sometimes they can cause an advantageous trait. All right, and those are the mutations that actually get passed down to the next generations and can lead to things like evolution and changes within the species. Okay, but we'll talk more about that with evolution. So this is duplication, extra copies of genes. It could be two sequences like here, it could be just one sequence, could be copied once, could be copied 3, could be copied 4 times. But duplication, doubling, copying of the gene segments. Inversion, remember to invert means to flip, to reverse. Okay, inversion, here we have our chromosome A, B, C, D, E, F, G, H, I, J. Segments G, H, and I become inverted. They flip. Okay, and now that order I, H, G is different. Okay, and believe it or not, just switching the order is enough to change how the genes function, the proteins that they produce, and obviously the functions that they carry out. All right, so inversion is reversing the order of the genes within the chromosome. A translocation is a little more complicated. Okay, translocation, a lot of people get it confused with crossing over because we're swapping gene segments from one chromosome to the other. But notice here, it's with non-homologous chromosomes. Therefore, the gene segments that are being swapped are not the same. Gene N and gene HIJ contain different traits, different characteristics. They're not supposed to be on different chromosomes. And when that happens between non-homologous chromosomes, it can lead to negative effects. Okay? So this first chromosome is supposed to have HIJ afterwards. Now it has N. This one has HIJ. That's not correct. Okay? And that's considered a mutation, a translocation. They translocate. Location is where they're placed. Trans means to move, so they move locations. They switch locations from one chromosome to a non-homologous, non-similar chromosome. Notice how this chromosome is originally shorter, this one's longer. They're not homologous. This is not just the copy from mom or dad. This could be some of the same, dad chromosomes switching DNA, which is not good. And last but not least, we have a deletion mutation. In a deletion, one gene segment or several gene segments are deleted. Okay, deletions involve the loss of chromosomal material, and actually this is one of the more common chromosomal mutations. Leads to many of the genetic disorders that we experience in the human population. Okay, segment C being deleted here, so instead of A, B, C, D, E, A, B, D, E. So we're missing a section of the chromosome, which obviously when you're missing DNA, you're not producing some of the proteins that you need, and you're going to be missing some traits that could be necessary for life or just for normal function within the life. All right, so this PowerPoint has been on mutations. Remember, they are not always a bad thing. We focus on the disorder part of mutation. All right, but remember the difference between a gene mutation Okay, and a chromosomal mutation. Gene mutation is the difference in nucleotide sequence. An adenine, a guanine, a cytosine, a thymine have been added or switched or deleted. And a chromosomal mutation is when entire genes, thousands of nucleotides, have been added or switched or reversed or deleted, or even an entire chromosome in a non-disjunction. Okay, remember to be checking your YGK sheet for the key concepts from this PowerPoint, making sure you can answer those questions, and please come to class tomorrow prepared to discuss these topics with your teacher. Thank you and good night.